हिमतीनं जगणार,
जीवनाचा धडा घेऊन हिमतीनं जगणार.
सावित्रीचं माहेर युक्तीचा आधार, ग्रंथांची रस आणि ज्ञानाची आस. सावित्रीचं माहेर युक्तीचा आधार, ग्रंथांची रस आणि ज्ञानाची आस. युक्तीचं शिक्षण हा देशाचा मान.
SBDM आटपाडी, SBDM आटपाडी, SBDM आटपाडी, SBDM आटपाडी.
गावात मुलांचे थवे म्हणती हेच कॉलेज हवे. कोणी भावड्या म्हणे कवड्या, कोणी लाजरा, कोणी बुजरा, कोणी हसरा, कोणी बितरा. आई बाबांना मुलांची धास्ती, पण पोरांच्या नसात मस्ती. पाठ लावून जिंकत कुस्ती. कॉलेजात आला न बदलून गेला, कॉलेजात आला न बदलून गेला. सरांच्या शिस्तीन सरळ झाला. आयडेंटिटी, ड्रेस कोड, असाइनमेंट हवे. शिक्षणाचे प्रयोग इथे रोज नवे नवे. गुरुला वंदन, मैत्रीला स्पंदन, मनाला बंधन, अभ्यासात मन.
SBDM आटपाडी, SBDM आटपाडी, SBDM आटपाडी, SBDM आटपाडी. एकीची शक्ती डोक्यात युक्ती, एकीची
आता दुकान यायचं तरी फक्त
मायेचे कोंदण नात्याचे बंधन विद्येचा सागर ते जीवनाला आकार SBDM आटपाडी SBDM आटपाडी SBDM आटपाडी SBDM आटपाडी संस्कार कष्टानं होतील स्वप्न ही साकार कष्टानं होतील स्वप्न ही साकार अंगाराला कधी लाचार जीवनाचा धडा घेऊन हिमतीनं जगणार जीवनाचा धडा घेऊन हिमतीनं जगणार
सावित्रीचं माहेर युतीचा आधार ग्रंथांची रस आणि ज्ञानाची आस सावित्रीचं माहेर युतीचा आधार ग्रंथांची रस आणि ज्ञानाची आस युतीचं शिक्षण हा देशाचा मान
SBDM आटपाडी SBDM आटपाडी SBDM आटपाडी SBDM आटपाडी
गावात मुलांचे थवे म्हणजे हेच कॉलेज हवे कोणी भावड्या म्हणे कवड्या कोणी लाजरा कोणी बुजरा कोणी हसरा कोणी बितरा आई बापाला मुलांची धास्ती पण पोरांच्या नसात मस्ती पाठ लावून जिंकत कुस्ती कॉलेजात आला न बदलून गेला कॉलेजात आला न बदलून गेला सरांच्या शिस्तीन सरळ झाला प्रयोग येते रोज नवे नवे. गुरुला वंदन, मैत्रीला स्पंदन, मनाला बंधन, अभ्यासात मन.
SBDM Atpadi SBDM Atpadi SBDM Atpadi SBDM.
Good morning, all of you. Myself, Professor Prabhakar Jadhav, Head Department of Botany, SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi. Once again, I welcome all of you for this one-day international e-conference on recent trends in life sciences. Firstly, I would like to welcome the chairman of the Atpadi Education Society, Atpadi, Mr. Amarsiya Deshmukh, for his inspiration to organize this one-day international e-conference. I also welcome Dr. S.M. Karande, the Vice President of the Atpadi Education Society, Atpadi, my teacher and former principal of this college, Srimant Babasaheb Deshmukh Mahavidyalaya, Atpadi. I also welcome acting principal of this college, Dr. Vijay Londe, who is always supporting us to organize such conferences. I also welcome today's resource persons, Dr. Jay Kumar Chavan from Yashwantrao Chavan Science Institute, Satara. Second speaker, Dr. Gajanan Ghode from South Korea, Dongguk University. I also welcome the organizing committee of this college and technical support. Again, this seminar or this e-conference is successful is due to the participants from the different countries. In this e-conference, recent trends in life sciences, we have registrations from 9 different countries, including India, as well as from India. There are near about 27 states which are included in this one-day international e-conference on recent trends in biodiversity. Totally near about there are 100 and more participants for this e-conference. So I welcome all the more than 1,000 participants, as well as today's guest resource persons, the chairman of the society, vice president of the society, principal of this college, resource persons, and all participants. So again, I welcome, and I stop my welcome speech. Now I request acting principal of this college, Dr. Vijay Londe, for his few words. Dr. Londe, sir, please.
Okay. Hello, everybody, and good morning. It gives me a great opportunity that today the Atpadi Education Society's SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA, Department of Botany, and IQAC jointly organized International E-Conference on topic recent trends in life sciences. I'm very glad because this is the first international e-conference which is organized by Department of Botany. I'm very much thankful to Head of Department Botany, Professor Prabhakar Jadhav, Professor Prakash Hasbe, Dr. Dhananjay Lohar, because we are very happily, gladly welcome to all participants, resource persons. Today's e-conference, we have 9 countries, including India, and 27 states of India, near about 1012 participants joined with us. And our technical team, our organizing team, organizing committee is very gracefully organizing this e-conference. For this e-conference, we have great speakers, very eminent alumni of this college. First, Dr. Jay Kumar Chavan, who is working Yashwantrao Chavan Institute of Science, Satara. And second, Dr. Gajanan Ghodke, who is working Dongguk University, South Korea. The Atpadi Education Society was established in 1953. And our college was established in June 1969. In 2019, we celebrated our golden jubilee year. Our science college celebrated silver jubilee year. Today, this conference subject recent trends in life sciences. So, dear participants across the globe, on the occasion of this international conference, I wish to convey the message that it is the need of the time to think and review our approach towards the global environment. The global leaders should change the way of thinking of global environment. The situation has arrived now where we all have to change our approach towards nature. The pandemic of COVID-19 is a signal given by nature to all of us. I think this is a wake-up call for all of us. We all have to review the way we all are continuously pressure on Mother Nature. We all must go hand in hand to save our Mother Nature by living eco-friendly lifestyle. We have to minimize our need, then only Mother Nature will survive and we will also. I request all dear participants across the globe to pass on this message that will surely help the coming generations to live a healthy life. Once again, I welcome our resource persons, and mostly I want to tell all of you, our great supporter, Honorable Amarsiya Deshmukh, is supporting to us this type of programs, these types of e-conferences. My humble welcome to Amarsi DESHMUKH, and second, our guide, our great leader, who is a former principal and now who is working very eminently, vice president of the Atpadi Education Society, Dr. Subhash Karande. I welcome to all of you. I thankful to these eminent personalities. Once again, all participants from 9 countries and 27 states in India, I welcome to all of you. Have a good day. Thank you.
Thank you, sir. Now I welcome the former principal of this college and vice president of this Atpadi Education Society, Dr. Karande sir, to inaugurate this function with some words. Karande sir.
Good morning, fellow attendees and participants. I am very happy to note that the participants from 9 countries, 27 states, and from India, about 1,000 participants are registered for this international conference. This is Dr. Subhash Karande, Vice President of the Atpadi Education Society Atpadi and the former principal of SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi, to be online with all of you for the inauguration of the International E-Conference on Recent Trends in Plant Sciences, which is jointly organized by the Department of Botany and IQAC of this college. I welcome all of you for this e-conference. Let me thank Dr. Vijay Londe, acting principal of this college, Dr. Shivaji Vibhute, chairman IQAC, Professor Prabhakar Jadhav, head department of botany, Dr. Dhananjay Lohar, convener of this e-conference, Professor Prakash Hasbe, organizing secretary, Dr. Deepak Rajmani, chairman organizing committee, and the members of organizing committee for inviting me for the formal inauguration session of this international e-conference. I am indeed very happy to declare that this international e-conference on recent trends in plant sciences is inaugurated, and hoping it will help be beneficial to the participants in upgrading their knowledge. Friends, it gives me great pleasure and satisfaction to note that a NAC peer team has visited this college exactly 5 years back during July 2015 and re-accredited this college with B grade with CGPA 2.67. A science faculty of this college has completed 25 years on June 2018. This college has completed 50 years on June 2019. And the botany department has completed 25 years recently in June 2020. On this background, the organization of the international conference by botany department and IQAC is appreciable. This is possible because of a dynamic leadership of Honorable Mr. Amar Singh DESHMUKH, the Chairman, the Atpadi Education Society, Atpadi, and the coordination and cooperation given by teaching and non-teaching staff of this college. As this is the International E-Conference and main theme is on life sciences. Life sciences, as you all of know, includes plant sciences, animal sciences, microbiology, and biochemistry, biotechnology, biophysics, cell biology, molecular biology, genetics, physiology, ecology, and the systematics. The majority of current biotechnological applications are microbial origin and it's widely appreciated. The exploitation of natural microbial diversity for the identification and assay of known enzymes currently active field in microbiology. I am very happy to note here that today's resource persons both are of our past students. One of them is Dr. Gajanan Ghodke from Korea, and his main field is biotechnology. Gold biotechnology, blue biotechnology, green biotechnology, red biotechnology, white biotechnology, yellow biotechnology, brown biotechnology, violet biotechnology, and dark biotechnology are some of the branches of biotechnology. I am only concentrating on dark biotechnology, and it is a color associated with bioterrorism or biological weapons or biowarfare, which uses microorganisms and toxins to cause diseases, death in human livestock and crops, and one of the recent example is COVID-19. Professor Gajanan Ghodke is expressing his views on phytoremediation. And according to me, a bioremediation is an important field in life sciences. It includes a phytomediation, mycoremediation, bioleaching, bioventing, bioaugmentation, rhizofiltration, and biostimulation. In phytomediation, where the plants are used as a source to remove the pollutant from the pollutant sites. But there are some limitations in plants, and therefore the remedial capacity of plants can be significantly improved by genetic manipulation and plant transformation techniques. The second speaker, Dr. Jay Kumar Chavan, he will be expressing his views on a tissue culture. The tissue culture is a technique is widely accepted and used for plant sciences, forestry, horticulture, under in vitro condition, under aseptic condition, for the conservation of endangered plants, protoplast fusion and regeneration of novel hybrids, stress resistance, herbicide resistance, embryo rescue, transgenic plants, artificial embryo expressed by artificial endosperm and seed coats, that is through somatic embryogenesis. For all the recent trends in life sciences, it requires a diversification in plant species, and that is a biodiversity. India, 8% of world's biological diversity on 2% of Earth's surface one of the 12 mega diversity countries in the world. The plant resources in India comprise about 46,440 species, representing about 11% of world's known flora. About 8,000 species of flowering plants are medicinal plants, but only 150 of them are extensively used in indigenous system for medicines. About 320 species of wild relatives of crop plants are known to have originated in India, and therefore India is considered is one of the 8 Pavlovian centers of origin and diversification of cultivated plants. For example, a rice variety grown in India are estimated to belong to 50,000 to 60,000 land races. And this is the area where the trends are possible for the research because of diversification of this type of cultivated plants. Friends, every year, the plant sciences researchers, they publish their papers in different journals. The country leading in world scientific publication in science and engineering. No one knows how many scientific journals there are, but it is estimated that around 30,000 journals and 20 lakhs articles published each year. The US National Science Foundation has released data showing that 25,55,959 science and engineering articles were published around the world in 2018. India published 1,35,000 research papers in 2018, 1,87,000 research papers in 2019, and in science and engineering, and become the 3rd largest publishers in the world. The ranking of publishing research papers in science and engineering, and the first 5 countries are China first, US second, India third, Germany fourth, Japan fifth. In 2019, China 6,84,000, US 6,78,000, India 1,87,000, Germany 1,83,000, Japan 1,32,000. So based on these publications in science and articles, there are ranking in the world, ranking in India, which are like this. The life sciences articles, number of research papers published by India in 2019. The first branch ranking first in India is biochemistry with 4,948 papers, and world rank is 4th. Next, second ranking in India for molecular biology with 4,300, and world ranking is 6th. With 3,164 articles, India ranking 3rd, world ranking 3rd. Biotechnology with papers 2,697, ranking in India 4th and world rank 3rd. Environmental science with 2,324 papers, ranking in India 5th, ranking in world 8th. Genetics with 1,916 research papers, ranking in India 6th, ranking of world 10th. Systematics, ecology and evolution, total papers published 1,906, ranking in India 7th, ranking for the world 11. Biochemistry, genetics and molecular biology, total paper published 1,651, ranking in India 8th, world ranking is also 8th. Biophysics, 1,508 papers with ranking in India 9th and world ranking 3rd. Cell biology, 1,480 papers with ranking in India is 10th and world ranking is 9th. Animal science, total papers 1,244, ranking in India 11th and world ranking 9th. Ecology, 1,123, ranking in India 12th and world ranking 11th. Microbiology, 1,121 research papers, ranking in India 13th and world ranking 6th. Applied microbiology and biotechnology, total paper published 1,100, ranking in India 14th and world rank 3rd. Physiology, total paper published 885, ranking in India 15th and world ranking 12th. Now these are the papers published during January 19 to December 19, that is of 12 months. Now, these are the papers published in science and engineering. Now, if you consider the papers published in natural sciences, and this is given by the Nature Index. A Nature Index tracks a contribution of research articles published in 82 highly qualified natural science journals chosen by an independent group of researchers. And according to Nature Index, the first 5 countries, the paper published in natural sciences are first USA, second China, third Germany, fourth UK, and fifth Japan. 6th France, 7th Canada, 8th Switzerland, 9th South Korea, and Australia 10th. In India, as far as India is concerned, there are 10 topmost institutes where the Nature Index has given that contributed for the publication of research papers. And the first one is Council of Scientific and Industrial Research, New Delhi, CSR, CSR Laboratories. Second, Indian Institute of Science, India. Third, Tata Institute of Fundamental Research. Fourth, Homi Bhabha National Institute of India. Fifth, Indian Institute of Technology, Bombay. Sixth, Indian Institute of Technology, Madras. Seventh, Indian Association of Cultivation of Science, Indian Institute of Science and Education and Research, Calcutta. Ninth, IIT Guwahati. And 10th, IIT Technology. Now this will see the trends of research in India. The first one in biotechnology and the last one in the physiology. Friends, these are the trends in research, and I welcome once again all of you and thank the organizers for inviting me for express my views on the occasion of this inaugural session of this one-day International E-Conference on recent trends in life sciences. Thank you, all of you.
Thank you, sir. Honorable Vice President of this society, Dr. Karande sir, have taken review of total life science, and for his review, once again I am thankful to him. And now I request Dr. Deepak Rajmane to introduce the speaker of the first session, Dr. Jay Kumar Chavan. Dr.
I deem it my pleasure to introduce speaker of the first session, Dr. Jay Kumar Dr. Jay Kumar Chavan. Dr. Jay Kumar Chavan is an assistant professor at Yashwantrao Chavan Institute of Science, Satara. He is a coordinator of the Department of Biotechnology. He is also coordinator of Internal Quality Assurance Cell, Satara. He has played a pivotal role in the establishment of Department of Biotechnology in the college. He is a renowned scientist. He has published a number of research papers in the national and international journals of repute. He was awarded prestigious Young Scientist Fellowship from the Department of Science and Technology, Government of India. He is also research advisor for Nanyang Academy of Science, Singapore. He is working as an editor for 2 international journals and worked as a peer reviewer for more than 10 journals of international repute. So far, he has published more than 32 research papers. He has 5 book chapters to his credit. So today we have such an eminent personality. So without wasting much time, I hand over the charge to Jay Kumar Chavan. So over to Jay Kumar Chavan. Thank you.
Is it audible?
Sir, start, sir. You start, sir. Yourself.
Okay. Thank you, Dr. Rajmane, sir, for kind words. Good morning, everyone. Honorable Chairman of the Atpadi Education Society, Honorable BABASAHEB DESHMUKH, Vice President and mentor of ours, ex-principal of SBDM, Principal Dr. Karande, sir. Acting principal, instead, I could use the word active principal of the SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi, Principal Dr. Vijay Londhe, Chief Organizer and Head Department of Botany, Professor Jadhav sir, Coordinator of Internal Quality Assurance Cell, Professor Shivajirao Vibhute, Organizing Secretary of this virtual gathering, Professor Hasbe, Convener of this event, Dr. Dhananjay Lohar, my fellow resource person, Dr. Gajanan Ghodke from South Korea, all the participants, delegates, and all the components of this virtual gathering. Just we saw that short video clip and the photographs of the journey of SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi through their on the occasion of their golden jubilee year, and they have achieved a lot. I must congratulate all the stakeholders, all the teaching and non-teaching staff and management of the Atpadi Education Society for their efforts to achieve the goal they were decided while establishing the SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi. Well, in the current situation of this challenging time of COVID-19 pandemic, we have connected through the virtual gathering And probably if COVID-19 was not there, it would be possible to meet in presence at the organizing site. I can see from the last few months, in fact, since the COVID-19 pandemic is there, the internal quality assurance cell and various departments of Srimant Babasaheb Deshmukh Mahavidyalaya Atpadi, they are organizing a series of events to sustain or to improve the quality among the academics as well as the administration, and they have organized the several events during this pandemic time or during this challenging time. I could see that organizing of any event is not a simple task. For this purpose, every staff from the principal to the last man of tune, everyone should be proactive, and then only the success of any events get. And I can found that such a proactive leadership, such a proactive teaching and non-teaching staff is there at the Srimant Babasaheb Deshmukh Mahavidyalaya Atpadi, and that's why only such kind of organization is possible. Apart from this, the management personalities, president, vice president, chairman, they are also cooperative. Due to this only, the success of any event we can measure. So, my dear friends, today I have been asked to talk on the recent objects in the plant tissue culture or the recent advances in the plant tissue culture. As I see that there are some students of the undergraduate studies and the postgraduate studies, they have connected for this virtual event. So instead starting directly with the what are the updates and advances in the plant tissue culture, we will proceed with the certain basic parts of the plant tissue culture, and we will end the session with whatever the advances is there in the plant tissue culture and what are the applications of the plant tissue culture. So, dear friends, as we are aware that plant tissue culture technique is one of the important branch of the biotechnology or one of the important tool of the biotechnology, which avails the certain avenues for the propagation of the plants. Most of us, we are aware that for propagation of any plant, we need seeds, either seeds or the stem cuttings. However, there are several number of plants. They are not propagated simply by using the seeds or using the stem cuttings. So for that purpose, the plant tissue culture helps to propagate such kind of plants. In the plant tissue culture technique, we can use any part of the plants, including root, shoot, either nodes, internodes, leaves, flowers, floral axis. As Principal Dr. Karande sir mentioned that several aspects of the plant tissue culture. So we can use that every part of the plant, and we can propagate the plants in the large numbers. And there are reproductive limitations in many plants, as like in certain plants, limitations in the flower production. If flower is produced, there is no seeds. If seeds are produced, there are no seed viability. So such kind of constraints are there in the plants, and number of plants are becoming the rare, endangered, and critically endangered category. So that's why the plant tissue culture technique is a vital route to propagate such plants. In simple terms, plant tissue culture technique is nothing but the in vitro cultivation of the plants or the plant parts on the defined nutritional media with controlled conditions. For the propagation of plants, the conditions which are needed are the aseptic conditions. Microorganism free area is the most important task. Then controlled temperature, light irradiation, then photoperiod cycle of 16-hour photoperiod cycle, then humidity. So these parameters are necessary to propagate any plants through the plant tissue culture technique. This plant tissue culture technique is totally based on the two major characteristics of the living cell or the plant living cell. One is the totipotency and other is the plasticity. totipotency, which means the ability of any plant cell to convert or to develop itself into the entire organism or the whole organism. That is the most important characteristics of the cell. And one more important characteristic is the plasticity. Plasticity means adaptability of the plant cell to the changed environment. So based on these two characteristics, the plant tissue culture technique is based. If we see the how this plant tissue culture technique is originated, the first attempt was done by the Haberland, and which is done a major work or the pioneering work in the field of plant tissue culture. Hence, this Haberland is called as the father of plant tissue culture, and thereafter several scientists or the several research groups, they have done work on the plant tissue culture right from the right to the recent ones. So basic need for the plant tissue culture is the culture media. Culture media is nothing but a mixture of substances which provides nutrients for growing and multiplication of the cells and tissues under controlled conditions. As we found that the plant absorbs the water and mineral elements from the soil. So just we are providing whatever the mineral elements or the whatever nutrients they are absorbing from the soil. In the plant tissue culture technique, we are providing such kinds of nutrients through the chemicals. That is the difference between your soil nutrients and the media nutrients. So the composition of the plant tissue culture media plays a vital role during the propagation of the plants during the in vitro cultivation, because the specificity of the plants, every species needs different types of the different composition of the plant tissue culture medium, and it largely depends on the species to species, genotype to genotype, and even if you consider the single genus, and under this 5-10 species are there, even though that 10 species require different kinds of the media composition. So this media composition is very specific to the species to species. There are we see that there are two different types of culture media. We can bifurcate into the what we can say synthetic and natural. Synthetic means chemically defined. In this category or in this type of the culture media, we are awareing that what is the exact composition of the media, what is the exact concentration of the media, and in the natural one, we are not aware that what is the exact chemical composition of that natural media. As per the requirement of the experiments, one can also classify the tissue culture media into solid and liquid as per the requirement of the experiments. If we are not using the solidifying agents, then it is considered as the liquid. It is generally used for the cell suspension cultures, and the solid and semi-solid media are generally used for the shoot cultures or the organ cultures. So the history of the plant tissue culture media was started right from the White's medium, which was considered as one of the earliest medium, which was composed by the White in 1943. However, the media which is formulated in 1962 for the cultivation of tobacco plants, which was invented by the Murashige and Skoog, that media is universally used for the culture of the plants, tissues, and cells of every plants with their optimization. Thereafter, there are several numbers of plant tissue culture media are coming in the market, like Linsmeyer and Skoog, Gamberg medium, niche and niche medium. Nowadays, Knudsen medium is especially composed or formulated for the propagation of orchids, and now commercially, specific to the plants, banana culture medium, orchid culture medium, ginger culture medium, they are readily available for the propagation of specific plants. However, for the optimization of the propagation techniques, Murashige and Skoog medium is largely and universally used for in the plant tissue culture. If we see the whatever the ingredients of the plant tissue culture, if we see the inorganic salts, then which again bifurcated into the macro and micro nutrients, then carbon and energy source in the form of sugars, then organic substances like vitamins, amino acids, activated charcoal, antibiotics, and several other organic supplements, then growth regulators, solidifying agents, and pH of that particular medium. These are the general ingredients or the composition of the Morashige-Skoog's medium. Before formulating the or composing the media, one can know about the exact role of the macronutrients as well as the micronutrients they are playing during the growth and development under the natural conditions. If we see the macronutrients, macronutrients are nothing but they are required more than the 0.5 millimolar per liter. There are major macronutrients are the nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. As we could know, we should know, as we are knowing that nitrogen plays an important role in the chlorophyll and amino acid synthesis. Magnesium is the major part of the photosynthesis. Sulfur is important for the formation of nodules in the leguminous plants. So such kinds of roles of individual micronutrients we should know. Then and then only we will be able to formulate, we will be able to optimize a composition of the media for a particular plant species. Similarly, micronutrients are there: molybdenum, zinc, boron, copper, manganese, iron. They also, though they are required in the very less quantity or very less concentration, they required or they plays an important role in the plants during in vitro regeneration. So this is the general composition. I am not going to detail of the what is the exact concentration of the macronutrients and micronutrients in the Morashige and the Skoog's medium. However, the carbon source is very much essential in the plant tissue culture medium. And this carbon source is generally used in the form of sugars. Especially sucrose is more preferred in the plant tissue culture rather than, there are several other options are there: glucose, fructose, lactose, galactose, raffinose, and several other sugars are there. However, sucrose is the major carbon and energy source which is generally used in the plant tissue culture medium. Why the sucrose is preferred in the plant tissue culture media? This is only because of once we add these sucrose into the plant tissue culture medium, and then we autoclave process is done. After that, the sucrose is converted into the glucose and fructose due to the hydrolysis. And in the initial part of the tissue culture technique, the explants or the plant cells or the plant organs or the plant parts which are cultured on the media, they absorbs the glucose. And in the latter part of the regeneration, they use the fructose. So most preferably, sucrose is used as the carbon source in the plant tissue culture media. Then certain organic supplements are there which are also used for the boosting the regeneration frequency or the regeneration process of the plants. Generally, these vitamins and every organic supplements, they are endogenously produced in the plants. However, when we cut the plants or the explants and it inoculates on the tissue culture media, at that time the capacity or the of the endogenous production of the organic supplements is reduced. And to gain this capacity of the production of the organic supplements, it is necessary to supply the exogenous supplementation of the organic, like vitamins, then vitamins like adenine, inositol, thymine, and generally those are used in the range of 0.1 to 10 mg per liter. There are several examples of the vitamins which are commonly used in the plant tissue culture of the several plants like nicotinic acid, ascorbic acid, then folic acid, etc. Then amino acids are also produced inside the or the endogenously in the plant tissues. However, during the in vitro regeneration, it needs the additional support from the additional source of amino acids and casein hydrolysate, glutamine, asparagine. These are the certain amino acids which are generally supported the in vitro growth and development of the plants. Then certain phenolic acids are also used for the plants, especially the phenolic compounds, chloroglucinol, which is used for in those plants whose shoots are unable to root under the in vitro cultivation. So for this purpose only, the phenolic compounds are used for as a stimulants for rooting to the shoots in such plants. Apart from this known composition, there are several growth adjuvants, or what we can say the natural complexes, which are also added in the plant tissue culture media. And generally, this exact composition or the concentration of these growth adjuvants are not known. That's why it is are undefined in nature. There are several examples of the natural complexes like coconut water, fish emulsion, protein hydrolysis, tomato juice, potato juice. For example, we can take that the coconut water. We are not aware that what is the exact concentration of the water. In the latter studies or in the latter research, it is known that the coconut water is a very good source of the cytokinase, especially kinetin, what we call it as the 6-furfuryl aminopurine, which is the major source or the major part of the coconut water. However, initially it was not aware that what exact chemical concentration of these natural complexes have. That's why these natural complexes are called as the undefined in nature, and they can also added into the media, and they boost the in vitro regeneration frequency of the plants. That activated charcoal is very rarely it is used in the plants because in the woody plants or the tree species, while culturing into the culture media, at that time the tissues or the plant parts which are cultured in on the media, they secrete certain toxic substances into the media. That's why there is a chance of decaying of that plant parts. To detox whatever the toxins what there in the media, they can by using the activated. This is especially in the woody plant tissues or the tree species. Then use of antibiotics. Use of antibiotics are generally avoided in the plant tissue culture media. However, in such cases, if you see that in the pathology, there are two types of infections. One is the superficial infection and another is the systemic infection. So superficial infection we can easily remove using the surface disinfectants like mercury chloride, sodium hypochlorite. However, the fungal infections which is inside the tissue, that we can call it as the systemic infection. And for removing such infection, addition of very less concentrations of the antibiotics such as streptomycin and kanamycin is recommended for such systemic infected plant species. Then solidifying agents are played important role for making the solidity or giving the solidity to the culture medium. This depends on the what experiments we are going to do. If we have to do the cell suspension cultures or the liquid cultures, there is no need to add the solidifying agents. There are two types of solidifying agents. One are which are extracted from the natural resources, which is called as the agar-agar, which is extracted from the gracilaria, that is the red algae. However, nowadays in the market there are several types of synthetic solidifying agents are available, like gelatin, gelride, phytagel, methyl. These are the certain examples of the synthetic solidifying agents. And the making of the solidity or the making the solid media is the only when used because it provides a great support for the tissue while growing into the medium. And one more thing here about the last part of the culture media is the pH. pH is one of the important part which makes the which supports the media to get solid, and generally 5.0 to 6, this is the range which is used for the optimization of the pH of the any plant tissue culture media. Media sterilization, that also depend on the what volume we have prepared, and generally it is used generally it is autoclaved, or generally it is sterilized by autoclaving at 121 degrees Celsius at 15 lbs pressure. And duration of this autoclaving or the sterilization process is dependent on the what volume of media is there. Then the plant growth regulators, which plays an important role in the plant tissue culture technique. Every species or the requirement of every plant species is very much specific. As we are aware that there are 5 different classes of the plant growth regulators as auxins, cytokinins, gibberellins, then your ethylene, abscisic acid. However, cytokinins, auxins, and gibberellins are most commonly used plant tissue culture in the plant tissue culture technique. Generally, we are aware that the cytokinins plays an important role in the cell division and cell elongation. So, in the plant tissue culture technique, we use these different kinds of cytokinins like benzylaminopirin, kinase 6-purpuryl amino acid, amine, then thiodiazurine, for the purpose of shoot induction as well as the shoot multiplication. Then next class of the plant growth regulator is the auxins. Auxins plays an important role in the during the in vitro rooting of the shoots, and there are several examples of the auxins which plays an important role in the various rare endangered medicinally important plant species are the indole-3 acetic acid, alpha-naphthalene acetic acid, and indole-3 butyric acid. These are the certain examples which plays an important role in the induction of roots during the plant tissue culture media. And application of auxin and cytokinin ratio for induction of the what we can call it as the mass of cell, that is the callus, and several other groups of the plant growth regulators like gibberellins, ethylenes, abscisic acids, and polyamides. They are also used, but in very rare cases and very specific cases they are used in the plant tissue culture technique. Then what are the factors which generally affects the plant tissue culture technique? The first, we can see that the types of culture vessels. There are different types of culture vessels for different types of experiments of the plant tissue culture techniques. If we have to optimize the conditions, or if we have to initiate the cultures, then at the initial stage we can use the test tubes, glass test tubes as the culture vessels. However, if aim of our or the goal of our experiment is to enhance the biomass, at that time we can use the glass bottles or the plastic pots. Nowadays in the market, plastic pots are available as reusable and autoclavable. So we can use for the several experiments. Then there are the glass plates and the glass plastic plates, which are generally used for the induction of somatic embryos or the callus cultures in several numbers of plant species. And as per the industrial requirements, there are several numbers of or the several types of bioreactors which are now available in the market for enhancing the biomass of the shoot cultures or any other cell suspension cultures or the callus culture. These are the certain other factors which affects the plant tissue culture or the micropropagation, that is the plant part. On the basis of the experiment, our goal or aim of the experiment, we are using the explant or the plant part. The plant part which are using for initiating the plant tissue culture process, that is called as the explant, and which part we are using, that is also one of the important tasks. If we have to produce the disease-free plants, we need to use the apical or the meristematic tissues. Then if you need to produce the large number of plants, then one can use the nodal buds. As per the requirement of the experiment, there are different types of the explants are there. which explant we are going to use that affects the plant tissue culture process or the success of the plant tissue culture process. Then genotype, that is the genotypes and species. Then physiological condition of the explant, that is if we are using at the active part or the healthy part or the young part or somewhat old part, that is also success of the plant tissue culture also depend on this. The size of the explant, which growth regulators we are providing in the plant tissue culture media, carbon source. These are the certain factors which generally affects the plant tissue culture technique. There are different types of cultures that like organ culture, meristem, bird, callus cell. We are not going into detail because it requires separate lectures or the separate sessions for particular culture techniques. I just highlight the different types. Dear friends, now we are in the era of, or now we are living in the age of nanotechnology. And nanotechnology plays an important role. Probably next speaker is the very good expert in the field of nanomaterials, and he has very good publications and technology transfers in the nanomaterials. So these nanomaterials plays an important part of the our life. They plays a very important role in agriculture, in medicine, in pharma, in every sector. We see the nanomaterials plays an important role. Nowadays, the nanomaterials are also used in the plant tissue culture media for different purposes. They plays very important role at different stages of the plant tissue culture. As like, if we see, there are different types of nanomaterials like silver oxide, zinc oxide, titanium oxide, aluminum oxide, cobalt, and gold nanoparticles. They plays an important role in the plant tissue culture. If we have to disinfect our explant or the plant tissue, then silver as well as zinc as well as the titanium nanoparticles pigments, which reduces the whatever infection is there on our explant. So for disinfection purpose, we can use these nanomaterials. Then during the differentiation and the morphogenesis phase or the stage, one can use the silver as well as the aluminum oxide, then gold, cobalt, as well as carbon nanotubes, which are also plays an important role during the differentiation and morphogenesis of the plant tissues under controlled conditions. If there, as I told, there are many plants which are becoming extinct from the nature. And if we need to conserve such kind of plants, we need to produce genetically diverse or genetically different plants. Because if any plant have a same genetic composition, there might be chances of that plant becoming extinct from the nature. So if we produce the genetically variable plants, some of the plants will survive in this nature or the wild conditions. So production of such somacronal varied plants or the genetically varied plants, silver as well as the silver nanoparticles plays an important role. Then if you want to produce the secondary metabolites in the form of elicitors, we can use different silver nanoparticles, aluminum nanoparticles, as well as the titanium nanoparticles. So these plays an important role to accumulate in more content or more value or the more amount about the secondary metabolite production. While in genetic transformation, one can use the silver nanoparticles for the genetic transformation studies. So these are the several applications of the nanoparticles nowadays which are used in the plant tissue culture technique. If we see here in the first photo, you can see the microbial as well as the bacterial infections in the plant tissue culture. In the first slide, microbial contamination at early stage, while just after the inoculation, and the bacterial contamination just after the certain period after the incubation. If we add the certain nanomaterials or the nanoparticles in the plant tissue culture media at very low concentration, such bacterial contaminations will be reduced and uninterrupted flow of the tissue culture process will be continued. So there are such several applications of nanomaterials are in the plant tissue culture medium, which helps to boost the tissue cultural process of several numbers of the plants. If you see what are the ways of to produce the plants through the tissue culture technique, there are two ways, that is direct and indirect way of the production of the shoots. Direct way means whatever explant or the plant part we are using from the donor or the mother plant, and direct to this explant we got the shoots. That is the direct way. And in the indirect way, one can find that there is one more additional step, that is the callus culture. From the explant or whatever plant part we are using from the mother or the donor plant, and for that from that callus culture again we have to initiate the shoots and then roots and then into the complete plant. So this is the general overview, direct and indirect organogenesis of the plants. There are 5 basic steps which are involved in the plant tissue culture. That is the selection and preparation of the plants, establishment of the aseptic cultures, then shoot multiplication, then rooting, acclimatization, and transfer to the soil. So selection of, as I told that, selection of the explant, it is totally dependent on the whatever the objective or the whatever the aim of the experiment is there. Then preparation, there are different types of explants are there, nodal buds, then as I told in the first slide only. Then preparation of the explant as like tap water washing, then detergent washing, then surface disinfectant with the different kinds of what we can say there's sterilants like mercury chloride is there, sodium hypochlorite is there, bromine water is there. There are several options for sterilizing the explant. And most important thing is that after the sterilization, we need to do this process under the aseptic conditions at the in the laminar airflow. Because why in the laminar airflow? Because laminar airflow provides a aseptic or the microorganism free environment, because of laminar airflow contains the highly efficient particulate air, that is the HEPA filters, which don't allow the microorganisms, fungal spores, or the any other dust particles into the laminar airflow chamber, and which creates the microorganism free environment. And under this environment, one has to do the sterilization and further processes of the plant tissue culture. Then establishment of the plant culture, then shoot multiplication using different plant growth regulators. Then one more step is the in vitro rooting to the micro shoots which are generated in vitro. This is the most important and crucial step in the plant tissue culture. That is acclimatization and transfer of the plants to the soil. Because there at this stage there is most chances for the decaying or the death of the plants. Because in plant tissue culture, during incubation, we have provided 25 degrees Celsius temperature for the plants. And suddenly, if we take out these plants outside the laboratory, there might be a chance of heat shock and becoming simultaneously becoming the death of the plants. So to avoid this direct lab to land, there are several stages which are involved in the acclimatization. Like that, we have to remove the plants from the test tube, and we have to wash on the root. We have to wash the roots under the tap water. Then treat the tap roots with certain fungicides like bivestine. Then we have to transfer into the plastic pots or the cups which are having the planting substrate. There are different types of planting substrate or the planting material is there, as like coco peat, garden soil, sand, then vermicompost. There are different types of substrates which are used for the during the acclimatization or the hardening of the plants. And the optimization of this substrate is also one of the crucial steps in the acclimatization and the acclimatization process. Then success also depends on how gradually we are increasing or we are providing the light, humidity, and temperature, these parameters. So if we took out these plantlets from the laboratory, then suddenly we kept these plants to the 35, 40 degrees Celsius, there might be chance of the decay or death. So for this purpose, we have to slowly, as like if we take out from the 25 degrees Celsius, then we need to provide 27, need to provide 30, need to provide 32 degrees Celsius. Likewise, we have to adapt these plants from 25 degrees Celsius to the whatever outside temperature is there. So need to be taken care during this acclimatization process. And on this basis only, we can measure the survival rate or the importance of the plant tissue culture. Once we produce the plants through the tissue culture technique, it need to be assessed for its stability. Stability means what? Are these micropropagated plants or the tissue culture raised plants are similar to the donor plant or the mother plant? On the basis of, we can assess or we can compare these plants or the stability on three bases, or there are three parameters we can check the stability of these plants. First thing is that the morphological or the phenotypic characters. We can easily compare that whatever the plant height is there, maturity date is there, flowering date is there, leaf size is there. These are the certain phenotypic characters. On that basis, we can compare the micropropagated plants are as similar like the donor or the mother plant. However, if in many cases the tissue culture raised plants are only similar like their donor or the mother plant, and this is the main
is a technical issue. Please wait for 2 minutes.
Hello. Please wait for some time. There is a interruption in net connectivity. So please wait and cooperate us.
It's audible, sir.
Okay, sir. Proceed.
Okay. Sorry for technical interruption. Just we are seeing the example of the biochemical markers which are generally used in the assessing the stability of the plantlets. If you are taking the example of the ginger, ginger is known for its pungent compounds, that is gingerols or chagols. Where one can compare the whatever content gingerol or the chagols are present in the wild field cultivated plants as well as the tissue culture raised plants. This is one of the way to test the stability assessment. Then one more technique which are recently used in the plant tissue culture technique, that is the genetic stability assessment using the molecular markers. Molecular markers like random amplified polymorphic DNA, simple sequence repeats, and several other markers are available in nowadays for assessing the genetic stability. Here you can see that the similar banding pattern is observed as like which is observed for the mother plant. So this is the most important technique nowadays which are used for the assessment of the genetic stability. There are several applications of the plant tissue culture techniques. We are not going into details. However, we will see certain examples of the applications of the plant tissue culture. As the principal Karande sir mentioned that plant tissue culture technique is generally used for the in situ and ex situ conservation through the micropropagation, cryopreservation, and synthetic seeds. That is the major objective of the plant tissue culture technique. Then metabolic engineering, enhanced production of the active compounds, then artificial seed production, haploid plant production. And nowadays, this plant tissue culture technique plays an important role in genome editing studies and synthetic biology. That is CRISPR-Cas technology. Nowadays, it is emerged in the field of biotechnology. So, for that purpose also, the plant tissue culture technique is useful. We can take one example only, that is the conservation of the biodiversity. What biodiversity means as the principal current sir mentioned, that the variations among the individuals and the number of individuals at a place. If we take the example of the biodiversity of the Class Plateau is one of the famous internationally known site. It is also recognized by the UNESCO as a World Natural Heritage Site. If we see this site covers only 25 square kilometer area. However, this 25 square meter area harbors more than 400 plants. And these 400 plants we cannot observe at the same time. The characteristic feature of this biodiversity hotspot is the seasonal changes. If we see this lateritic plateau or this plateau in the month of during the time of summer, it is just a naked lateritic plateau. After the first spring, you will able to see certain ephemerals as well as some pogostemon, which we call as poticholi. And in the subsequent season, after 15 or 20 days interval, we will able to see the certain utricularia populations, ureocolon, then sorry, the smithia, and in the end part of the spring, you will able to see the different types of grasses. So this biodiversity spot mainly harbors over 400 plant species within the limited area. Then it shows 24% of the endemism, and most importantly, this Class Plateau gave 5 new species to the science or to the globe. So such biodiversity hotspots mainly consist of the rare, endangered, and threatened plants, which are generally conserved using the plant tissue culture techniques. If your bandwidth or the net connectivity is up to the mark, one can able to see this slide. That is how the seasonal changes occurs at the biodiversity hotspot or at the what we can say at the Class Plateau. This lantern flower is among the endemic, endangered, and critically endangered plant group, which is also conserved now by using the plant tissue culture technique. Because only developing a technique or developing a protocol is the not aim of the plant tissue culture. Whatever the plantlets or whatever the plants which we are generated or raised through the plant tissue culture technique, that we should have to reintroduce or the reimplant into the natural populations or the natural localities. Then and then only the purpose of the tissue culture will be served. Then we can also conserve the wild relatives of the crop plants. Wild relatives plays an important role in the breeding practices, plant breeding practices. And here we can take one example of the vigna. Most of the vigna species are the creepers, or they grow alongside the soil. However, there is only one species, which is the Vigna khandalensis, which is the very sturdy and erect species and the very disease-resistant species. So, such kind of genes from these wild relatives we can transfer to the cultivated plants, and we can make the disease-resistant varieties. Then other benefit or the application of the plant tissue culture technique is the production of secondary metabolites. And secondary metabolites plays an important role in the diagnosis of various diseases or the curing of the various diseases of the human beings. And one can obtain the secondary metabolites by using the shoot cultures, callus culture, cell suspension culture, then root culture. There are several ways, or the hairy root culture. For the enhancement of these secondary metabolites in the plant tissue culture, one can use the route of elicitation. Elicitation means the stimulation or the boosting production by using different types of elicitors. Like there are 2 types of elicitors, that is abiotic and biotic elicitors. Then biotic elicitors, one can use the fungal strains, then hormones, bacterial strains, then saccharides. And in case of the abiotics elicitors, one can use the chemicals or the physicals like UV radiations or the changing light intensity. There are several numbers of the plants products which are secondary metabolites which are developed using the plant tissue culture technique, like artemisinin. then Taxol is there, Mangiferin is there, several number of secondary metabolites which are produced using the plant tissue culture technique. As we are aware that artemisinin is a very good anti-malarial agent, and which was invented by the Yu Tu, that is the lady from the China, and she got the Nobel Prize for this inventory of the artemisinin from the Artemisia. Okay, this is the list of the several numbers of the secondary metabolites which are generally produced using the plant tissue culture technique and elicitation process. Now we will end up the session with the whatever work which is currently going on at my side. Currently, we are doing the developing the protocols for the elite clones, production of elite clones of the Salacea chinensis, because these Salacea chinensis clones are very much useful and very much are the chief source of the mangiferin, salacinol, and cotylenol. Generally, those are possesses anti-cancer, anti-obesity, and anti-HIV properties. So these plants collects from the wild localities very illegally nowadays, and that's why we are producing the alternative ways for the production of these plants by using the tissue cultural techniques. And last part is that the certain wild gingers we are now cultivating in our area by using the plant tissue culture techniques, because the wild growing gingers are which lacks the pungent compounds, and by using the plant tissue cultural technique and elicitors and precursors, we are enhancing or we are stimulating these pungent compounds in the wild gingers, and nowadays we are providing these plant lists to farmers also. And this investigation or this study is well supported by the various funding agencies like DST and UGC. So overall, this is the plant tissue culture technique which is now used in the various sectors like agri, pharma, clinical, and food everywhere, which plays an important role. So at the end, I should extend my sincere gratitude towards the organizers for providing this opportunity to express my views among this virtual gathering. Dr. Lohar sir, it's over to you. This session is now open for discussion. If you have any queries, then you can put into the chat box, or you can raise your hands so organizers will able to connect or unmute you.
Thank you, Dr. J.
Okay, if there are no doubts or no queries, it means it is 100% percolated or nothing has been percolated among the participants.
Jay दिसत नाही का त्याला?
Yes, sir.
Thank you, Dr. J.
Yes, sir.
There are no questions from participants.
Okay.
Okay, so
I think,
thank you.
I think Dr. Gajanan sir have some query. He wants. He's raising his hand. Please unmute him.
गजानन सर बोलतील तर त्यांना बोलू. झालं बोलायचं?
Hello.
Please, Dr. Godke.
Yes.
Ask your query.
Yes. Thank you, Dr. Jaykumar. Nice presentation. Actually, I am interested in anti-cancer plants. So,
Okay.
are you working on anti-cancer plants to propagate because they are under threat of extinction?
Yeah, currently I'm working three five plants which are
Okay.
so already proven the anti-cancerous activity. As like
Okay.
Salacea chinensis, there are 5-6 species are there from the northern Western Ghats. So definitely we will collaborate for this.
Yeah. That's great. Yeah. Thank you.
Okay, so all queries have covered, and now I request Dr. Dhananjay Lohar for the vote of thanks of the first session. Dr. Lohar, sir.
Thank you, sir. This is the first international conference organized by our department and IQAC. For this first session, Dr. Jay Kumar Chavan was the speaker. He expressed his view very enthusiastically, and he explained how tissue culture is very much useful for the our life. Thank you, Jay Kumar. And now I hand over the screen towards the head, Professor Prabhakar Jadhav, to have next session.
Thank you, Dr. Lohar, sir. The first session is over. Now we are proceeding towards the session number 2, which will be conducted by Dr. Gajanan Godke. So I request Dr. Shivaji Vibhute to introduce Dr. Gajanan Godke.
Yeah, please, sir, please disable the screen sharing.
Thank you very much, Professor Prabhakar Jadhav, sir, for giving me the opportunity to introduce the second speaker of this international conference based on the recent advances or recent trends into the life sciences. It is a great opportunity to me to introduce this speaker. The second speaker for this session is Dr. Gajanan Godke. He is working as an associate professor in the Department of Biological and Environmental Sciences, Dongguk University, South Korea. He has 94 publications to his credit with 2,263 citations with H-index 26, Citation index 45. His contribution to research is very huge, but I have just sorted out few things. The area of his research work is on the treatment of hazardous engineered nanomaterials, adsorptive remediation of cobalt oxide nanoparticles, magnetized and chitosan functionalized alpha cellulose fibers, spectroscopic determination of iron, green synthesis of silver nanoparticles, and so on. And he has invited talks at different places around the globe. Now it is very important that the topic today he is going to speak on is recent advances in phytoremediation. With this brief introduction, I welcome Dr. Gajanan Godke sir to speak on this topic. And very important mentioning is that he is the product of Shivaji University, Kolhapur, and he is the alumni of this parent institution, SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA, Atpadi. He is there into the Dongguk University for last 12 years, working on in the research. He is a postdoctoral fellow, worked there, and now has joined as an associate professor into that university. Now, with this brief introduction, once again, I welcome Dr. Gajanan Godke sir to speak on his topic of interest, and that is recent advances in phytoremediation. Dr. Gajanan Godke sir.
Yes, thank you.
Hello.
Hello. Please continue, sir.
Yes, can I connect first? Google Drive documents.
You are audible. Please start your session.
Yes. Thank you. Can you play the PPT? Hello. Can you play the PPT? Yeah, thank you. Good afternoon, good morning, good afternoon, all of you. First of all, I am very thankful to the organizing committee of the International Conference on the recent trends in life sciences. I am also very thankful to the Honorable Amar Singh Bapu Deshmukh Sahib and Dr. Subhash Karande sir, Professor Vijay Londe sir, Professor Prabhakar Jadhav sir, Professor Prakash Hasbe sir, Professor Dhananjay Lohar sir, Professor Shivaji Vibhuti sir, Professor Deepak Rajmani sir, Professor Jay Kumar Chavan, and Science Faculty of SHRIMANT BABASAHEB DESHMUKH MAHAVIDYALAYA Atpadi. I congratulate organizing committee and technical staff and the leadership of the college for their efforts in organizing e-conference in COVID-19 pandemic crisis. I warmly welcome all students and participants. I am very delighted to be here and present recent trends in the phytoremediation. I hope researchers and students will find some research opportunities in this field. This presentation will provide a brief review on recent progress in research and applications of phytoremediation techniques to protect our mother nature. This presentation will cover interdisciplinary topics related to plant-based remediation, technologies, microbial community, ecology, environmental protection. Remediation is related to environmental protection. So if it is based on the plant, we can call as plant-based phytoremediation. There has been increasing interest in phytoremediation because the use of plants can be used to reduce concentration of pollutants, or it can make to reduce the harmful products into the harmless. Let's start with the 4 major type of phytoremediation mechanisms or strategies we can call that are predominant in implementing on-site remediation. So the first of all, I would like to present about the 4 different types. So the first one is phytoremediation. So can you go to the first slide? So please select the first slide. That is about the phytoextraction. So there are 4 type of methods: phytoextraction, phytostabilization, rhizodegradation, and phytovolatilization. And the first one is the phytoextraction. That is about the extracting heavy metals, because we cannot degrade the chemical elements that are heavy metals. So we can just extract using the plants. So So let's continue. This can be done by the cultivation of metal tolerant. There is some pop-up. What is this? Okay. So please select the first slide and second slide. So we will continue here. Here we can use the many different type of metal tolerant plants that are especially used for the extraction of metals. So we can call them again the hyperaccumulators of the metals. So these are the very well-known plants for all of us. So we will consider some of them in the detail in the later. That are the Indian mustard, sunflower, amaranth. That is the we can the Marathi call as Tandraja, and Ipomia. These are the very prominent varieties that has a hyperaccumulator kind of property. So once we accumulate these heavy metals from the soil or using some aquatic plants, so it gets in the vegetative part. So this is one of the most important that we can use these plants to extract the metals. And this process also we can call as a sorry. So can you select the first slide, please? No, not possible. Again, the first one, yeah. Okay, next one. Okay, and phytostabilization. Second mechanism is the phytostabilization. So unlike phytoextraction, the phytostabilization focus mainly on sequestering pollutants, that means limiting their dispersion from the particular place. So this process can be used to limit the all kind of pollutants from the place where they are present. So we can avoid the risk of infiltration. And this mechanism allows a very significant possibility to reduce the toxicity of the pollutant, because it gets the cover by capping, bonding by some of the biopolymers, proteins, antioxidant compounds. So this has less toxicity as compared to the original one. So this has to be used to reduce the transfer of the pollutants from the contaminated site to the food chain. So it can be not completely assured, but we can just get that possibility, but it is not make sure that it is going to be avoided. So we cannot assure that. So understanding this mechanism, however, it is not yet well established in scientific literature. So because this process is quite complex and it has been not well explored yet. So let's consider the third most important mechanism is the rhizodegradation. Please go to the next slide. Rhizodegradation. Yes, thank you. So in this process, we can call it this, how it can biodegradation can happen with the help of microorganisms to break down the hazardous pollutant. And this process occurs in the rhizosphere, the area where the soil surrounding the roots and the plant. So the microbial activity and the plant, they collectively work together and perform the rhizodegradation. So this is about the root and the plant, roots and the microbial community, they play the very significant role combined together. So we can call it this is symbiotic process. So we can stimulate this process using some kind of the nutrients, adding some kind of bioaugmenting agents. So this process can be stimulated to grow the bacterial or fungal community and to make the more source. So microorganisms such as yeast, fungi, bacteria, algae also can be involved here in this process. So it can be stimulated in the microbial and rhizosphere position. So addition of nutrients are must to grow the optimal level of the microorganisms. So this can be called also the phytostimulation in addition to the rhizosphere biodegradation. So this is a very synergistic system, because when the plant releases the natural carbon containing substance and such as sugar, and that can be used for the microorganism as a nutrient, and it stimulates the activity and it performs very well to degrade the many kind of organic nutrients. So let's go to the next slide. Yes. So here we can consider the bioaugmentation. This is the one of the most important process occurs always in the natural process. Bioaugmentation is kind of biological process where it can be done by addition of some rhizobacteria, microbes, fungi, or it can be happens as a naturally. So we can collect some of the samples from the outside, and then we can study their activity if they are very useful because they are effective, if they are very tolerable because they are found from that site. So we can multiply those microorganisms and we put them back to the sites. So these organisms originated from the contaminated site are being focused. But in addition to this, there are many well-known microorganisms that can be also used to perform this bioaugmentation process. Indigenous varieties have a very effective metabolic capability to perform remediation process. However, the other species also can be used to sophisticate the very specific pathways to degrade a particular contaminants. So there are very positive outcomes from this process of bioaugmentation, such as improvement of efficiency and increased speed of the breakdown of the contaminants and reduce the toxic matter in the system. So let's move to the next slide. So we can look for how we can go for the soil amendment approaches. Please go to the next slide. It's about soil amendment. So suppose we are performing phytoremediation activities in the soil. So if it has to be performed at optimum level, we have to provide the sufficient number of nutrients. So this is the very effective environment friendly approach for the contaminated soil as well as infertile soil cleanup. So here we can use, please go to the next slide, please. And this process use organic and inorganic nutrients. Those are helpful to improve the physical, chemical nature properties of the soil. So the range of the natural materials, we all know that agro forestry waste, agriculture waste, compost, like industrial waste, like some kind of compost, manure, molasses has been used for this enhancing the phytoremediation practices, but some of them are not recommended because of their chemical content and possibility of the additional contamination of the soil. But here I would like to mention that biochar is the most potent and effective material that can be used to enhance the bioremediation practices. Again, humic acid and some kind of plant extracts also has been reported as a promising agent for the as a soil amendment and enhancing the phytoremediation practices. So far, biochar material is the most effective because in addition to the soil amendment, it helps because it's nature to help the remediation of both organic and inorganic nutrients. So these are the one of the two strategies we can use in the enhancement of the phytoremediation. So let's move to the next slide about the phytovolatilization. So phytovolatilization is one of the most significant process for remediation of the groundwater and remediation of the volatile compounds present in the soil. So this process is a very unique as compared to the other process. So all these four processes are very unique and different. So the most important thing is that this process is a very cost-effective, environment-friendly approach to use in the remediation of the volatile compound. This process occurs when the plant is growing. Plant absorbs the large amount of water, and in addition to this organic contaminant. So as water travels through the root and leaves, the vascular system takes the all contaminants and either convert them or modify or transport them into the safer and directly release this volatile compound into the air. So we can call this process a very effective transpiration. So some of the plants are very effective in this. So we will discuss which plants are very effective. So here we can consider some of the trees species, woody tree species. They go straight to the down and collect the groundwater and convert into the volatile compound and turn into the and that finally that products get converted into the safer because UV radiation in the air convert that volatile compounds or degrade that compounds. So we will discuss which plants are very effective in the phytovolatilization. So let's move to the next slide. There are many different contamination sites. Contamination is throughout the world. There is huge contamination sites are appearing these days. Let's move to the next slide. And this phytoremediation technologies, how it can be applicable to different type of contaminated sites like air contamination, water contamination, or contaminated sediments, groundwater contamination, wastewater streams like industrial, agriculture, municipal, sewage. There are many kind of wastewater streams. Please move to the next slide. And this contamination term itself suggests that we can fix the contaminated sites according to we can restrict restricted place where the soil or water contamination has been confirmed. Yes, move to the next slide. This is one of the high severity of the impact of the ecosystem on human health as a remediation. So these are the different source where the land, water, air are getting polluted from the industrial, because this we cannot see the visible, but even though excessive use of the agriculture, some kind of pesticides, chemicals, they are finally end to the wastewater streams, and they comes to the groundwater, and finally we get exposure. I would like to give example that is where in the Punjab state has been used a huge amount of the pesticides, and now that is the reason that large number of percentage of the people now getting the cancer and other metabolic disorders because of this excessive use of the. So we can not only consider industrial, municipal, there are huge amount of the waste, like particularly in our areas, like Eastern country produces huge amount of the textile waste, water waste. So it is very visible to all of us. So can you imagine how much groundwater has been polluted by the organic and inorganic pollutants present in the such wastewater that has been directly discharged in the freshwater river sites. So this is one of the serious concerns where we will look how we can look for this application in the specific problems. So let's move to the next slide. There are two types of inorganic pollutants, the organic and inorganic. So these categories are very significantly affecting our environment. So environmental pollution caused by metals, metalloids, inorganic nutrients, even we can consider that is excessive use of NPK and sulfur and phosphate in the agriculture practice, as well as from industrial waste. Radionuclides these are the inorganic pollutants are now visible in agriculture land in the nearby the industrial waste, and the domestic wastewater has been deposited there. So there are several types of organic pollutants, and some of the organic pollutants are very persistent, or it can be called as recalcitrant. They are not easy to degrade, like DDT and like persistent organic pollutants. They stay in the soil or water for year and year, and they affect the human and animal health as well as aquatic plant health. So these are the very different categories has been present in the different source of the wastewater as well as the landfills. So currently there are great efforts have been made to reduce, to remove or stabilize various kind of contaminants present in this kind of polluted sites. So let's look for the which systems we can look to address the particular kind of problems. Let's move to the next slide. So here we will consider hydroponic system, constructed wetlands, floating wetlands, planting trees, and farming polluted lands. So let's go by one. So these are the different systems we can use. See here, I have shown here the illustration schematic diagram of phytoremediation treatment, how we can perform in the laboratory scale. Like suppose selecting one of the contaminant like heavy metal, single heavy metal or single organic compound, and we can perform this experiment in the laboratories using the aquatic plants like Centella, Apomia, Salvinia, Pistia, Eichhornia, and many more we can look for. And once they are effective, then we can look for the constructed wetland, how they can move to the large scale. So the phytoremediation technology is using the living plants to clean up soil, air, and water contaminated with hazardous contaminants. So this technology has been proposed as a cost-effective plant-based approach for environmental remediation. The concentrating effect of the result from the ability of certain plants, like they have just concentrating effect of the contaminants. So these are called as hyperaccumulator, as I told you before. So can be used to bioaccumulate chemicals. We can extract those chemicals and we can safely dispose. So let's move to the next system, next slide. So we can look for the hydroponic systems. So these are also possible in the laboratory settings. These hydroponic systems, I will discuss two types of system, water culture and NFT. So these two culture, these are very sophisticated small setup in the laboratory we can develop and work on the individual plant, individual contaminant, how they are effective for cleanup or degradation. So here in the system, it is very, very important that water should be well oxygenated and sufficient with the nutrients. These are two important critical factors. So without oxygen supplement, this process cannot be provided, and both micro and macronutrients are needed to survive and to thrive the plants and to work for the degradation. So we should make sure that when we use such system in our research laboratories, the roots should be matter should not be submerged in a nutrient solution. It just to be exposed, the root system to the nutrient solution, no stem and no vegetation. Otherwise, the plant will die. Just a root exposure. So the water will be continuously directly to there is no need of any supplement of the water cycle. So let's move to the next system, that is need to pump the water and to feed the plant, and that water come back to the system. And this is again same system. We can use this system for growing lettuce, like vegetables, and some kind of tomato plants, or some many different kind of vegetable plants to grow this system. And the same system we can also use for our experimental studies. Using this just one setup for a triplicate time, we can perform a single experiment at the time. So this is very effective laboratory-based setting for small scale as well as large scale. That is, there is no limit. So this is the very sophisticated setup. Everything material is available. We can buy and just set up, and we can use it. Let's go for the constructed wetland possibilities. Let's move to the next slide. So here I would like to discuss 4 different kind of setups we can use in the constructed wetland, because it is in the natural as well as in the constructed. It can be process occurs in the both. But when we construct, we can use these models. So the first model is free floating plants. We know that plants floats freely and they take care of the wastewater. But the height of the water should be not much, otherwise that will be not effective. Another is that free water surface constructed wetland with emergent plant species, as well as subsurface with horizontal and vertical, the last two one. So this way, these two systems, there is the flow of the wastewater, there is outflow of the treated water. So these are the constructed wetlands. We can employ the microphytic plants growth in this system. So there are many different aquatic plants. Those are microphytic plants. They are very effective in utilizing this microphytic aquatic plants growing nearby water. They may be either emergent with upright position portion or above the water surface, like submerged floating. So these are the different examples using the microphytic species, including Thypha, Hydrilla, water hyacinth, or duckweed. So there are many more species also we can explore, but these are the very common species has been used in this constructed wetland systems to treat the hazardous wastewater in the nature as well as in the artificial form. So let's move to the next slide. So how about the using this constructed wetland for the nitrification? Nitrification is the one of the most significant cycle in the nature that happens in the naturally. But when we are going to process the large amount of the waste water that is having the ammonia in the water, it is from the maybe mostly animal sources as from the industrial sources. It has been treated well, otherwise it is very harmful to the aquatic plant as well as animals and fish. So how this process occurs, this is the combination of two type of bacteria. Nitrosomonas bacteria, there are various species works in the system. Nitrobacters, they are placed to convert ammonia into the nitrite, and finally second step after that nitrates. And after that, that becomes nutrient for the plant, and that plant can grow, particularly with which are the aquatic plants. So here, the most critical point is the oxygen in the present water concentration should be high enough to support the growth of the strictly aerobic nitrifying bacteria. And another is that nitrification can be coupled with denitrification. So this denitrification should be avoided using the optimized process. So this can be performed for the nitrogen removal. Let's move to the next slide. This is also another system is like constructed wetland for just volatilization, because these are not conversion, but just volatilize the ammonia in the air. Ammonia comes from the livestock or manure or some of the industrial waste. In this system, we can use the high concentration of the plankton, that is periphytic algae. So again, here the algae also play the major role in the phyto because it comes in the plant category. So, but where the water surface, where the algal growth assemblage can create the high pH value during the daytime of the photosynthetic activity. Because of this, that allows to volatilize significant amount of the nitrogen from the constructed wetland to the open surface, where objective of this process also use can be for the monitoring the volatilization of the ammonia. Next, please. Yes, it's very visible that how we can employ the aquatic plants, different aquatic plants. Those are growing differently. So that particularly here we can discuss the static plants as previously discussed. This has huge opportunity to generate the expanding aquatic remediation, because this has huge ability to produce a large amount of biomass. We can periodically harvest the biomass, and we can treat or we can use for various other purposes that are not in the food chain. So we can provide such ecosystem services once we study about the potential phytoremediation technologies to recover biomass very effectively. Freshwater plants are used for such purpose. So let's move to the very advanced next slide. Technique has been recently become very popular. This was about the static plants. Let's move to the next. Yeah, sure. Here, let's move to the discuss about the free-floating biofilms. This is recently developed a very advanced technology. You can see in the image, the plant uptake the accounts about the 10%. That means plants are just providing the habitat to the microorganism, and the actual process has been not performed by the plants, but the habitat is been created over there. So the microbial community thrives, and they take the huge part of the treatment of the water that is naturally happening. So the root, what they helps to the grow very deeper and act a very important surface area for microbial growth. So they form biofilms. We know that biofilms, that means the bacterial or algal also, there are both possibilities, because in the aquatic system, they assimilate together and they form the film. So what happened here, they work for removal of nitrogen, as we discussed, removal of ammonia and phosphorus through the microbes. That can be done immediately, because that makes the better environment for the plant to grow. In cooperation, the plant awards the food carbon source for the microbial activity. So this is the symbiotic relationship, boost efficiency of cleaning by the both participant, and this is the floating wetland. So this is a very effective method to perform phytoremediation on the island, on the pond, as well as in the dams, or in anywhere there is wastewater problems. So let's move to the next. So I would like to discuss about one of the case study I was performed during the PhD time. I used 3 types of species: Brassica, sorghum, and Phaeolus, to just check, because before I was knowing that Brassica is very effective, that is Indian mustard, because that is hyper accumulator tolerant. So I just compare, I just selected other. So there are some significant differences between these other 2 species and Brassica, about the enzyme, about the effectiveness of accumulation of the heavy metals from the textile wastewater, as well as decoloration of textile wastewater. This was one of the topic in my PhD time. I used this system in detail, and that research was published in the International Journal of Phytoremediation. So here, this is the very significant problem in our area, and the rivers nearby the Sangli and Kolhapur, these are the mostly polluted, and this is big threat to the people who are living there, the animals and the habitat. The habitat has been burned out. Just you can see the image. there is no possibility of growing any other vegetation once the exposure of this. So this is a very severe problem, is not yet considered very significantly. This textbook water also contain recalcitrant dyes that are not easy to degrade. So there are many different problems in the textile wastewater, and the most important that the textile industry use huge amount of water in the processing of their textile products, and there is unabsorbed the color directly goes to the, and some of them treating well, others are just disposing. So this is one of the serious. So let's consider how we can select specific species of agro or forestry species that can are very effective to solve the contaminated lands. So let's go for the next slide and discuss about the which agriculture crops, but they are not recommended for the food purpose. They are strictly should be under regulation. They should be used either for fuel, fiber purpose, fiber crops, and that should be not used for any edible purpose. So there should be chance of the adulteration of the food chain, but that should be under very carefully. So the rapeseed, that means canola, mustard, amaranthus, sunflower, Indian mustard, grasses, some of the grasses can be cultivated either in intercropping or monoculture. It should be under rotation. So remediation option using phytoremediation, soil particularly, it can be intercropping means we can plan with trees as well as this species. So let's go for Indian mustard and sunflower in detail. So the next slide is discussing about the significant use of the Indian mustard. So let's move to the next slide. Yeah, okay, here. So Indian mustard has been very effective as a metal accumulating plant, and this can tolerate even toxic metals. Here we can use organic matter, organic chelating agents, some of the fertilizers to support the plant growth in the contaminated sites. This can be aesthetic tourism if we take care of some protection about the health and safety. It can be also for the large scale if we are doing. It can be also for the value addition of the aesthetic also. This can be like tourist place sometime if that is not harmful to the direct exposure to the, because if there is no volatile contaminant, there is no harm to just walk on the such land. So these plants can be collected and we can process and safely dispose. So it will not come. So it takes time. Maybe such remediation of contaminated site take years of practice and making sure that the level of the contaminants is reduced below the acceptable level. So this is one of the most important. This plant has been worldwide used for heavy metal contamination. So let's move to the next slide. Another most effective plant is sunflower. Sunflower is also a hyper accumulator for the heavy metals. It can tolerate high amount of high percentage of metal content in their tissues. Some tissues are very inactive, so that doesn't damage the plant. So there are many different mechanisms, antioxidant, enzyme base, some of the polymer base, because we can see the white polymer in the sunflower. So this deposition of the metals once extracted in the plant tissue goes to the non-active part of the plant. Again, this plant has been known for stimulation of osmotic system, also helps accumulation of the plants. This plant also has been widely adopted as a phytoremediation practice, particularly for metal contaminated soils. This species have huge capacity to produce high biomass production as well as metal accumulation. So we can consider where we can use such biomass very carefully for the very selective purpose, not the all kind of purpose. So let's look for the next I would turn now intercropping, cropping from the agriculture to the agroforestry system. So when we are going to address different problems, we have to look for agroforestry. So this system has known to improve the efficiency of the phytoremediation very effectively. In addition to this, these woody plants can be intercrop with some of the agriculture crops as we discussed now, or some other like grass species. Those are very adaptable or local. Herbaceous species that who grows in intercropping can be also selected. Some nitrogen fixing bacteria also can be, plants can be also used for, because they can also generate the nitrogen, and it can, the collective like land-based symbiotic process can be also work. We can call like cooperation by intercropping. Let's move to the next. We will discuss agroforestry. Consider here the two major agroforestry species. Let's move to the next slide. So here we will discuss the agroforestry. So the woody plants like the poplar and willows, those are very popular, very effective, and there have been many different species have been found. And some of the hybrid plants, as Karande sir told, those has been developed, those has ability to tolerate broad diversity of the contaminants. And another most important effectiveness is these plants are effective not for the only one problem of the environment, but the multiple problem. So because they perform the phytoextraction, phytostabilization, rhizodegradation simultaneously. So they are not performing individual, but this is happening. So in addition to this, these species, poplar and willow, the poplar genus is Populus. There are about more than 25 species are well known. That grows very big trees, and the willows are not big trees, just small trees that are more than 400 species available. And these both species are known for rapid growth. Within a year, they produce tons of the biomass. And these poplar are so very ideal for biomass production. And so using this bio-based phytotechnologies such as phytoremediation can be very effective, not to solve just one problem, but many problems. We will conclude in the later slide. Let's move to the next slide to talk about the significance of the willow salix species. So this willow species, so these are been traditionally used for from the historical time. I don't know how the people know that in that time, because there might be not much pollution, but this has been grown worldwide. Consider here, there are options like physical, chemical, biological, that means phyto or biodegradation. So these are the, suppose make three categories. So the physical comes like excavation, transport, and landfilling of the contaminated sites. But ultimately that is not going to solve. This will be the just take the problem from here and reach the problem to another place. So the excavation, transport, or landfilling, these are very expensive processes, and that has been just performed for a particular reason. Suppose like if there is a residential area and if there is a possibility of the leakage or infiltration of the contaminants, that has been immediately excavated. Otherwise, mostly not recommended. However, phytoremediation using willow in particular thought to be very sustainable, very effective, green, and sustainable approach. And it is ultimate, the final solution for these problems. So this will clean up everything. So there is no residual contamination left over. These willows are also associated with microbial communities, various kind of communities. So this is again cooperation, symbiotic phytoremediation that makes very successful. And the willow trees can grow in high acidic condition like pH 3 to pH 4. This is another advantage for it. Can be used for heavy metals. There are two important varieties, Salix sejournia, Salix feminalis are the best producer of wood biomass, and it has been known that it can produce it's about 3 tons of solid biomass per hectare per year. So this woody biomass can be processed using different technologies available for energy production, raw material for biorefineries. So this is a very cost effective as compared to the restoring the mining sites and as compared to the landfills. Another most important thing is that suppose if we use chemical technologies to remediate, so finally we can use the coagulation, some case catalysis. These are when coagulation is done and that is separated from the water, that pollutant has to be treated on the site using some bacterial, fungal, or plant-based biological process. So it can be combined, but the major role can be played by the biological, and the other technologies like physical and chemical can be integrated together to make successful. Next, move to the next slide. So here we will discuss the significance of the popular genus Populus. So you can see the photo of the how tree. It can be for most of the people also just plant for the timber also. And these plants are very unique as compared, and they are very well known that they act as water pump. They grow deeper, the roots go deeper in the water level, that groundwater, and they transpires enormous amount of the water. So at the same time, they can also take the contaminants to the water that goes to the vegetative part, leaves, and finally to the, maybe there being possibility of the modification, degradation, transformation, and volatilization. This process occurs together. So this all process works. And this plant roots are known to create the ozone that is above groundwater level in order to obtain high transpiration rate. So this position, like ozone, is the position at which the groundwater, where the water in the soil pores appears because of the atmospheric pressure. So that level the roots grows inside. So because of high transpiration rate, they are totally dependent on the groundwater. So there is no need of extra care of watering and nutrient or amendment, soil amendment, no need. So they work there. So what we can do after performing this, like decontamination or remediation of the land sites, because the many municipalities are interested to establish to treat their land sites, the waste site, using this plant technologies. So finally, when we have the treatment like the wastewater, groundwater, land, what we can do this fast growing trees. Biomass can be used like paper making, industrial pellet, biofuels, paper, pulp, and even for the musical instrument, if they are very safe for their contaminant level, if there is no any residual. So the look here, and in addition, I have not considered the advantage that plant-based technologies, like growing the large scale technology, like plant-based. So there are series of advantage, like global warming and oxygen, and another is like this purpose, like environment protection. So huge benefits this remediation technologies. For example, these technologies, cost of the phytoremediation is lower as compared to the traditional processes for the both at in situ and ex situ settings. So the possibility of the recovery and reuse of valuable metals, if they are valuable, otherwise it cannot be used, can be used for phytomining as well as if there are very precious metals. We can preserve the topsoil, maintain the fertility of the soil. We can improve the quality of soil, increase the soil health, yield of the plant, and plant phytochemicals. These are the so many different advantages. Some plant can be used for to reduce soil erosion, metal leaching in the soil. So these are the so many different advantages in addition to the phytoremediation. So, but there are some limitation. So these are the research opportunities. You must found some of them in this presentation. But if I count some of the limitation of this technology, you can also look, is it possible to solve these limitations? Like phytoremediation has limitation because it's up to surface area, how much they are not going to solve the or other surface. So it is specific to the surface area and a depth occupied by the roots. The contaminants in the deep root, the deep soil that has been to be selectively particular species. And it is not possible completely to prevent leaching. As I told you that phytostabilization, it is just a process happens definitely, but it is not assurance that it is going to be prevent complete leaching of the contaminants from the groundwater or from the water anywhere. So we cannot be rely on those processes, but we have to also consider other technologies and process. So another most important problem is the survival of the plant get affected because of the pH, the contaminant, and the toxicity of the to the plants. So this has the factors that has to be considered. So the bioaccumulation of the contaminant, especially the metal, they can affect the consumer products. So it is strictly avoided. It can not be used for cosmetic purpose, food purpose, or any kind of the fooder purpose. So it is strictly prohibited. So it can cause a serious problem. And there is need or require safe disposal of the affected plant material. So these are some of the limitations. So we can consider those and work on those. So if we could develop great technologies to address, because the industrial pollution is enormous and is widespread, ubiquitous to the not one nation or many other countries also. So there are great opportunities to develop even technologies or services based on this phytoremediation understandings. So it can be very grateful to be here. I'm very happy and very thankful to the organizing committee for giving this opportunity. Thank you so much. And please ask questions. Just feel free and ask whatever you have questions. Thank you.
Suggestions?
Yes, please. Yes, Karan sir. Yes. Singh Parihar. Any question? Pratiksha Bhatt. Any question? Yes. Yes, please. Any question?
That we are looking for the questions.
Yeah, Karani sir. I expect question from Karani sir. Karani sir, can you listen me? Are you? I am audible to you. Hello. Yes, Faisal Islam. Currently, sir, no. Jay Chavan. Yeah, Jay Chavan has raised his hand. Please, go ahead. Yes, Jai Jawan.
Hello.
Yes, Dr. Jai Kumar.
Yes, Dr. Godke, really it was a wonderful session.
Yeah, thank you.
Yes, I don't have any query actually. Just I want to compliment you.
Yeah. Yes, Jai Kumar Chavan. Can I know your question?
Yes, yes, sir.
What is question?
Just network problem, Dr. Godke.
Okay, okay. Take time.
I will connect you personally later on.
Anyone have questions?
Hello.
Raise your hands.
Yes, Jai Kumar has question, but he is not connected.
Hello.
Yes,
I am
Karande
Dr.
sir.
Patel.
Yeah.
Yes, Suhas Kamble sir.
Yes.
Sir.
I want to ask one question. I'm
Yes,
working
sir.
on the wastewater treatment in Kurkum MIDC, where there is wastewater having high salinity, and it has got a color also. And in that, trying to grow some of the plants, but due to high salinity, these are not growing. So which
Salinity.
plant species? Which plant species?
For salinity, we have to look for the plant. Those are growing nearby the ocean. Those plants are very very established, very well established in the some
mangrove,
kind of
side with
plants.
mangrove.
Like I cannot recommend like coconut, but coconut
No,
grows
mangrove.
nearby. But some other species has to be explored from
Mangroves
the
I have tried.
ocean sites. Yeah.
Yeah. Okay.
Some has been collected and then try to grow and propagate. That may be work. If there is a high salinity, that can be work. But please avoid the dispersal, like storing the wastewater in the safe tanks and safe ocean. Otherwise, it can be percolate in the groundwater. That should be first.
So
for
we have taken a
aquatic plants,
precaution.
for aquatic plants, we have to go to the water bodies where the high salt level is there.
Okay, thank you.
Yes, Jai Kumar.
Khambe sir,
Yes.
please ask your query. Please cover, then I will call Dr. M. Tamil Selvan. Dr. Selvan, M. Tamil Selvan sir, please ask your question or queries.
Dr. Tamil Selvan.
Hello. Dr. M. Tamil Selvan.
Yeah. Sir, I am in the line only.
Yeah. Please ask.
Sir, can you hear me?
Yeah, sure.
Yes, sir. Sir, in our recent years, so in which way these biotechnological tools will be useful for the farming community, sir? As a farmer's point of view.
Yeah.
Yes, sir. As a farmer's point of view, sir. Not for our academic purpose.
I see.
So academic purpose, we have different role to play, sir.
Okay. So what is your question?
Sir?
What is your question?
Yes, sir. Sir, in our recent biological researches, biotechnology play a major role, sir.
Yeah, sure.
So this using this biotechnological role, which way it will be useful for the farming community, sir, as a practical?
Okay. Yeah. I understand your question.
Yes, sir.
Actually, the science technology is
Yes, sir.
a very great leap.
Yes, sir.
But the knowledge transfer from the science research and development, then it is missing. So the most of the farmers are usually go to the maybe kind of exhibition.
Yes, sir.
There are some technologies that comes like after commercial purpose. So the real understanding knowledge that implementation for the agriculture purpose.
Yes,
my
sir.
humble request, whoever the farmers in your contact, please ask them for go for diversity, not one culture. Go for multi-layer, multiculture, and because these are the some models has been developed by agriculture universities, but farmers are not ready to accept because they have been traditionally doing monoculture,
Yes, sir.
same cropping year and year after. So even sometimes they are not doing rotation like sugarcane. So
Yes,
it is a serious threat to the like our agriculture land to get infertile in near future.
Yes,
So I think it's a serious concern that has to be immediately accepted. It's a great for a farmer security, and without this there is no farmer security.
Yes, sir. Yes, sir.
One crop has been like that can be go for the depth.
Yes,
that
sir.
cannot solve any problem. So I also am doing my level best, but according
yes, sir. Yes, sir.
using this platform,
Yes, sir. Yes, sir.
not inform to you, but
Yes, sir.
everybody.
Yes, sir. Sure.
As we motivate and teach, and once the convince them.
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
They can practice. There are many different models.
Yes, sir. Yes, sir. Yes, sir. Okay,
so
okay.
That can be great opportunity. That can be timely plan. One farmer should have at least 50 vegetable varieties.
Yes, sir. Yes, sir. Sure, sir. Sure, sir. Yes, sir. Yes, sir.
Yes, and 50 fruit varieties.
Yes, sir.
Food security as well as for his income also. Because this has been ignored. This has been traditionally done, but these days has been turned. Because we can know that in the our recent past, there has been planting that like tree species, fruits near the house, garden, and then vegetable. They grow every type of vegetable and they store and they use for year long. That
Yes,
was
sir. Yes, sir.
real security, but it is missing nowadays. So
Missing nowadays. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Sir, thank you very much, sir. Thank you, sir. Thank you. Thank you.
Yeah, please. Anybody?
Okay. I request to all participants.
No, sir.
Request to all
Karande sir want to
participants
ask something.
that if
Karande sir
anyone
want to
have
ask
any
something.
query, they can
Karande
ask
sir.
or
Karande
raise their
sir,
hand.
Yes,
Dr.
sir.
Karande sir, please use continue. Dr. Karande sir.
Hello. Hello,
Yes,
Dr.
sir.
Ghosh.
I can listen to you.
Thank you for your nice presentation and information about phytoremediation. I would like to ask one question.
Yes, sir.
Is there any remedy regarding the nanotechnology? Have you any
Yeah, there is
information
remedy.
about?
There is remedy, but there every chemical technology, nanotechnology, all have limitations. Suppose that are based on catalyst, that are not universal, that are not work for all different kind of. But one plant can take care of several heavy metals, several contaminants. But one catalyst cannot take care of all those problems. So this has severe limitation. These technologies, this work for specific compound to convert them into the safer or to they make them some byproducts. They definitely work. There are nanocatalysts recently well explored. Those are being used for particularly for organic pollutants like textile wastewater, industrial wastewater, and pesticides, micropollutants from like those like metabolic disruptors. So this compound has been successfully degraded, but this has to be recycled, repeated use. There is chance again, suppose we apply directly to the nature, there is chance to the even contamination by nano contamination. This is the recently, as I think you might gone through my recent publication. I'm also working on the contamination by nano material because these are the severe many industries are manufacturing nano material using for different products. So there is chance to get the nanomaterial into the environment. So what is the fate of the nanomaterial and how we can protect the environment from the nanomaterial threats in near future? Yeah. Thank you.
Okay, thank you, sir.
I think there are no questions from the participants.
Yes, sir.
There are no questions from the participants. Thank you, Gajanan, for nice presentation.
Jay Chavan is in contact? Is in network?
No, Jay Chavan will ask you personally.
Yeah, sure.
We have told like that.
Okay.
So, to conclude this session,
yes, sir.
For the vote of thanks, I call Professor Prakash Hasbe for the vote of thanks of this one-day international e-conference on recent trends in biodiversity.
Thank you, sir. Thank you.
Thank you, Zazo sir. I am here to express vote of thanks of this international e-conference on recent trends in life sciences. First of all, I thank all the participants from different corners of India and across the globe. I thank both the eminent speakers for accepting our invitation and enlightening participants with their talks. I am also thankful to Chairman of Atpadi Education Society, Honorable Amarsiah Deshmukh, Vice President of Sanstha, Dr. Subhash Karande sir, for gracing this e-conference. I am thankful to Dr. Vijay Londe, Acting Principal of this college, who is a driving force in the conduction of this e-conference. I take this opportunity to express my heartfelt thanks to my colleagues from Department of Botany, IQAC coordinator, members of technical committee, members of organizing committee, and one and all. Thank you, sir.
Okay.
बंधन वितेचा साखर ते जीवनाला SBDM Atpadi SBDM.
Hello. Hello. The feedback link will be sent within 30 minutes.
It is already shared on chat box.
Shared in chat box and YouTube live streaming.
It is already shared in chat box and Telegram. So please submit your feedbacks and get your certificates. Those who are not going to submit the feedback, they will not get certificate. Thank you for registration and all process. Thank you very much.
Thank you, sir. Can I go?